Hello everyone and welcome to The Light Show, otherwise known as the Analytical Spectroscopy Technology Forum, organised by the Analytical Scientist and supported by Wytech, Brooker Optics, Hamamatsu Photonics Europe and DRS Daylight Solutions. My name is Rich Whitworth, I'm the Content Director at the Analytical Scientist, and I'm delighted to host our fourth Technology Forum, which will shine a light on the wonderful field of spectroscopy by bringing together 4 application experts from some of the world's top manufacturers. Before I pass you on to our speakers today, I'll offer you a quick glimpse of what's to come. First, Miriam Boehmler, Director of Applications and Support at Wytech, will explore confocal Raman microscopy and its powerful ability to offer spatially resolved chemical characterization in life sciences, pharma and beyond. Tom Tague, an Applications Manager at Brooker Optics, will put Raman Spectroscopy into your hands, literally, with a portable analyzer that delivers benchtop power. Moritz Fisher, who's a sales engineer with Hamamatsu Photonics Europe, will delve into the nuts and bolts, or perhaps the lasers and detectors of Raman optical systems. And finally, David Nichols, a senior applications engineer at DRS Daylight Solutions, will brief you on quantum cascade laser technology and the advanced measurements it enables. After the 4 short, but I'm sure highly informative presentations, we'll be opening up a live Q&A session. So please remember you can submit questions at any time throughout the presentations. All of the speakers are here for you, so it'd be great if you take advantage of their expertise. If you have a question that's specifically for one of our speakers today, if you can make a note of that at the beginning of your question, that would be great, and it will make my life much easier. Okay, that's enough from me. Let's get this light show properly on the road by handing you over to Miriam Boehmler, Director of Applications and Support at Wytech. Miriam, please take it away.
Hi everybody, thank you for attending this forum today and for joining my presentation now. My name is Miriam, and within the next 15 minutes I want to give you an idea of the analytical power of confocal Raman imaging and its correlation with other techniques. For those who are not familiar with Raman yet, let me briefly introduce what it is and what is it good for. The Raman effect is based on the inelastic scattering of light by the molecules of gaseous, liquid or solid materials. The interaction of a molecule with photons causes vibration of its chemical bonds, leading to specific energy shifts in the scattered light. Thus, any given chemical compound produces a particular Raman spectrum when excited and can be easily identified by this individual fingerprint. Raman spectroscopy is a well-established, label-free and non-destructive method for analyzing the molecular composition of a sample. And this is how our confocal Raman imaging and spectroscopy system, the Alpha 300R, looks like. Now, almost all kinds of materials can be investigated with Raman spectroscopy and microscopy, and thus there is a huge variety of applications, like in pharmaceutical industry, material sciences, environment and pollution, in food and packaging materials, life sciences, archaeology and forensics, new energy, and many, many more. Today, we will concentrate on pharma and life sciences, and I will present some examples that demonstrate the power of Raman imaging as an analytical tool. The first example is from scientific study by the group of Molly Stevens in UK, which they published in Nature Communications in 2020 in open access. In there, you find a fascinating study on living zebrafish embryos. So when we talk about Raman imaging, it can be much more than just one-dimensional spectroscopy. In fact, with our innovative technology, it is straightforward to acquire 2D or even 3D Raman images. The basis for such an analysis is a system that provides a combination of the highest spatial resolution, the highest detection sensitivity, and the highest measurement speed, which then allows you to obtain, for example, full volumetric biomolecular images. And I want to stress an important point here. Raman spectroscopy is label-free. Raman scattering is an intrinsic property of materials, and thus no labeling or sample staining is necessary. But let us go one step back. As I mentioned before, the Raman spectrum is like a sample's unique fingerprint. The easiest way to identify an unknown material by its Raman spectrum is by comparing the spectrum to a database of spectra of known substances. VTech invented TrueMatch for this purpose, an integrated Raman spectral database management software, and this is how it looks like. The red spectrum here is the Raman spectrum from that unknown particle that you see here in the zoom-in. The best match from the database, shown in blue here, then tells us that this is a paracetamol particle, and the additional information from the database is shown on the top right section. As you see, Raman spectroscopy allows you to chemically identify a material. Next step is clear. What about the analysis of not one single one, but many particles? And the solution is the Particle Scout analysis tool, which allows for the automated analysis of tens of thousands of target particles. In the example shown on this slide, an analgesic, that is painkilling, and antipyretic, anti-fever powder sample was investigated with Particle Scout by first acquiring a dark field image that you see here on the left. From this image, the location and morphology of all particles is extracted automatically by the software. Criteria were then selected that focused the further analysis on particles with a maximum diameter of less than 100 micrometer and an area of 1 square micrometer or larger. This highlighted a total of 3,052 particles. A Raman spectrum was then acquired from each particle in an automated way, followed directly by the chemical identification with the integrated TrueMatch Raman spectral database software. The measurement revealed that the majority of the particles were the analgesic agents A and E. Caffeine was present as an effect enhancer, lactose as a carrier, and white pigment was also detected. A quantitative report describing the proportions of the substances found in the sample and their distributions in relation to size was then generated. This example shows how particle properties and quantities can be correlated with their chemical composition. In case you investigate bulky samples instead of powders, you will need two-dimensional Raman imaging in order to analyze the distribution of specific compounds or phases. So let me now show you how Raman images are measured. My example here is hand cream with a moisturizing ingredient. On the left, you see the bright field microscopic image in which we see some bubble-like structure, but yet we have no idea what these bubbles are. Of course, we can measure the Raman spectrum at specific positions to identify the components. At the blue position, we find an aqueous phase with a very broad peak at the right end of the spectrum, which indicates water. The red spectrum, measured on one of these bubble-like shapes, is an organic phase. And the green spectrum, measured on another bubble, reveals Raman peaks that are characteristic for urea. And now we can apply Raman imaging to get the full picture. This is done by defining the size of the measurement and the pixel size indicated here by this grid. The idea is to measure full Raman spectrum at each individual pixel. Let's start the measurement. From the information in each pixel's Raman spectrum, it is straightforward to create a Raman image that shows the distribution of the different components. Image pixels in red color indicate positions where we have the organic phase without urea. Green pixels show the organic phase containing urea. And the blue pixels show the distribution of the aqueous phase, which forms the matrix in this emulsion. Pixel by pixel and line by line, we obtain the full Raman image, which now gives us the chemical information with a high spatial resolution. Another example for a 2D Raman analysis is shown on this slide. You see the bright field image of an immobilized red cell, which has been prepared in a buffered solution. The Raman image on the right side then provides the chemical composition within the cell. Obviously, Raman microscopy is capable of visualizing the composition, but also changes therein. Macrophages are known to play a key role in atherosclerosis via uncontrolled and excessive uptake of lipids, which transforms them into so-called foam cells. The goal of Matthäus and his colleagues was to investigate the lipid uptake in individual cells and better understand the contributing processes. For this purpose, they incubated the macrophages with different deuteride lipid species, such as fatty acids or cholesterol, and measured Raman images over several hours to observe the uptake dynamics and to visualize the intracellular distribution. This Raman image shows a cell after 30 minutes incubation with a fatty acid. A few lipid droplets are visible in red color. The rest of the cell is kept in blue. After 3 hours, the number of new lipid droplets has increased significantly, and again after 6 hours. Finally, after 30 hours, the Raman image revealed that the region of the cell around the nucleus is dominated by the lipid droplets. The macrophage is transformed into a foam cell. The authors demonstrated that Raman imaging can be a highly efficient and easy accessible tool for identifying cell-drug interactions and monitoring cell death pathways. Besides single cells, also tissue can be imaged. Tissue engineering, which is important, for example, in the development of biological implants, strongly requires non-destructive analysis tools, such as Raman Spectroscopy. And Raman Microscopy allows for the direct visualization of architecture and cell morphology in tissue. Tissue structures like collagens and elastic fibers can be detected and localized efficiently. For example, here you see the color-coded Raman image of a cross-section of a heart valve, revealing the cellular matrix components in different colors. Corresponding Raman spectra are shown on the right in the same colors. The collagen fibers are colored in red in this image, with the endothelium in yellow marking the outer interface. The elastic fibers that are embedded in the collagenous matrix are clearly visible in green color. This elastin-rich layer marks the blood inflow site of the valve. As you see, Raman imaging is suited to visualize and analyze structures in tissue in a non-destructive and label-free manner. It is further suited to investigate modification and changes in cells and tissue. So far, we have been looking at one-point Raman spectroscopy to identify materials, 2D Raman imaging, revealing their distributions, and of course, 3D imaging is also possible. To obtain a 3D Raman image, a stack of 2D images is measured, pixel by pixel, line by line, and layer by layer. And by doing so, we get the Raman information of a full 3D volume. The sample here is the hand cream with urea again. And this time we measured a full 3D Raman image. It consists of 600,000 Raman spectra. An integration time of 5 milliseconds per spectrum was used, leading to a total data acquisition time of less than 1.5 hours. And as the group of Molly Stevens demonstrated, this is even possible in vivo. They outlined broad applications of this method, not only in visualizing the biomolecular distribution of whole embryos in 3D, but also in resolving anatomical features at subcellular spatial resolution, in biomolecular profiling and discrimination of wild-type and mutant bacterium strains in zebrafish embryo model of tuberculosis, and even in vivo temporal monitoring of the wound response in living zebrafish embryos. The next logical step is to combine Raman imaging with other analytical tools. In the last minutes, I will touch some of these options that VTech offers in correlative Raman imaging. First of all, our Raman systems can be combined with scanning electron microscopes, for example, with TESCAN or ZEISS SEMs, to correlate chemical information provided by Raman with the even higher resolved information from SEM. It can also be combined with additional detectors like EDS, increasing the available correlative information significantly. During the measurement, the sample stays in the vacuum chamber, which can be a crucial requirement, for example, to study battery materials. The image you see here shows an inhalation particle, the Raman image on the left and the SEM image on the right. The integration of both methods provides a more comprehensive view of the sample by overlaying chemical and structural properties, thus linking morphology and chemical composition. Also, other materials can be investigated with rice. In this example here, a differentiation of bacteria by morphology and chemical composition is demonstrated. Two types of E. coli have been investigated, revealing differences in shape and also in the Raman spectrum, and allowing for the direct correlation. Also, a coated tablet has been analyzed by rice. For this purpose, the pill was cut and polished and then mounted in the SEM chamber with a piece of conductive carbon tape in order to avoid charging. The SEM image on the bottom left reveals areas of different morphology. The area top right is supposed to be coating. The bright field image on the bottom right looks completely different, indicating that it would be troublesome to find the same samples, but again, if the system was not fully integrated. Rice setups ensure that the same sample area is analyzed with both the electron and the light beam, making the experiment easy and straightforward. In the example shown here, the Raman image provides the additional chemical information, revealing the API in green, the excipient in red, and confirming that the top right area is the coating material. Also, fluorescence microscopy is a well-established method to visualize the insides of complex cell and tissue structures. Although intensive staining is required for fluorescence microscopy, it is sometimes desirable to correlate information of both Raman and fluorescence imaging from the same sample. Due to the modular design of the VTEC systems, this is easily possible on all platforms, exemplarily shown here for the inverted alpha 300RI system. And as example here, you see first the fluorescence microscopic image of some cell nuclei that have been stained with DAPI. The subsequent measurement of a Raman image reveals further unlabeled details. In this specific example, Raman imaging reveals the endoplasmic reticulum in red and the nucleoli in light blue color. And in addition, all setups are upgradable also with fluorescence microscopy, differential interference contrast microscopy, and phase contrast microscopy. So to summarize, I hope I could provide you with a nice overview of some Raman applications in pharma and life science. We have been investigating single particles with one-dimensional spectroscopy. We looked at tissue and cells with Raman imaging. You have seen that even 3D full volumetric measurements of samples are possible. You've seen that the identification, the chemical identification, by using this fingerprint Raman spectrum for individual materials is possible. You've seen that high-resolution localization of the chemical compounds is straightforward. Raman spectroscopy and imaging is label-free and non-destructive, and no alteration or damage of the samples is required. No specialized sample preparation is needed. In addition, I showed you some correlative techniques like the rice, which is the combination of Raman and SEM, as well as the combination of Raman and epifluorescence on biological samples. So next, I'm looking forward to answer your questions in the Q&A session or anytime via email. Thank you very much for listening to this presentation.
Thanks so much, Miriam. Great to see YTech, a serial winner of the Analytical Scientist Innovation Awards, still pushing the limits there. Okay, next up, Tom Tague will take Raman into the field with a small but mighty handheld device. Tom, the stage is yours.
Well, it's my pleasure to talk with you today about the Bravo in the Analytical Lab. The Bravo is a handheld Raman device that was designed really for a twofold purpose. The first is verifying knowns. So when you look at analyzing sample, there are two types of samples you would look to characterize. One is verifying your knowns or identifying unknowns. And so the workflow when we designed the Bravo was actually designed to optimize both acquisition types. So if you look in the center, you can see, if you look carefully, you can see that we can verify materials or we can acquire material and do the identification. So on the left, you see we can do raw materials verification and characterization, product quality compliance, distribution verification. And this is important because it can significantly affect dissolution. And then when we look also at identifying of unknowns, we can look at contamination, identification, product defect analysis, customer complaints, and maybe most importantly, reverse engineering, huge savings in cost for development there. So reverse engineering is maybe the most popular and most common type of application for the Bravo these days. So let's go through the first concept, which is essentially verification of knowns. So we have a barcode scanner, so you would scan that and then do the measurement, usually through the packaging material, and then it's just accepted red light, green light operation. Doesn't really get any easier than that. You would have your database of compounds that you would be looking to verify. And again, it's just red light, green light operation. You can also see that it's common for people to use gloves, sometimes big bulky gloves. We did receive the question in the past, well, did we need to have a screen that is that large? And actually, yes. You need to have the buttons large enough so that people can actually do work. And so I think that the button size in the software is actually optimal. Now, for identifying unknowns, we actually want to back up a little bit and take a look at Raman spectroscopy and see how it works a little bit. So what we're going to do is impinge high intensity monochromatic light, also known as laser light, onto a sample. And then we'll look to see what happens to the light. The light can be scattered elastically or can not interact with the sample. Those, as it turns out, are more common than the Raman transition that is desirable. And so you can also have the interference from fluorescence signals, and we want to make sure that we mitigate that. So we've actually designed the Bravo system, as you'll see in the next couple of slides, to actually really suppress fluorescence interference, which is really the biggest roadblock towards collecting good Raman data. So what is collected? Again, the backscattered light that is scattered inelastically, where the change in wavelength correlates directly to the Raman shift. And so we'll plot the intensity as a function of the Raman shift, and that Stokes shift is what we're predominantly interested in. Just remember your vibrational spectroscopy 101 basics is that each molecule has its own fingerprint signature in what we call the middle infrared region of the electromagnetic spectrum. That means that when you look at employing Raman spectroscopy in the analytical lab, this is a front of the line technique. It's non-invasive, non-destructive, and very, very specific. There isn't a more specific technique available. So it's just wonderful. Now, the results are stored on board, and you have the databases stored on board, and so you really don't need to do anything else. We do have many art museums that use the Bravo routinely, and they many times will put it onto a tripod and then use a wireless connection to a PC to do the acquisition, because they don't want to be moving the Bravo around by touching the screen. So it can operate wirelessly through a tablet or a PC, or directly as a device-only operation. Again, it's non-destructive, non-invasive, no sample prep needed. You can go through packaging materials, which is really good. So essentially all you need is a packaging material that is transparent to Raman, and again, that works great. Now, when we developed the Bravo, one of the things we really wanted to bring to the market was a tool that could actually get you around fluorescence. And so we've done that three ways. One is that, well, let's just use a couple of different lasers at the same time. So we have a duo laser configuration where we're using two different lasers. So if the electronic transition for fluorescence is at one wavelength, we can either go above or below that transition many times to actually get around the fluorescence. Very, very useful. We also actively control both lasers so that we can actually shift a few times around, moving the laser wavelength, emission wavelength around just a little bit, and then divide off the fluorescence, which doesn't really change with such small shifts. And lastly, we, in addition to the previously mentioned SSE that is patented, we also have the patented concave baseline correction for fluorescence removal. This uses concave or parabolic type functions to strip off fluorescence or scattering, again, which is most useful. And as we go through some of the features of the Bravo, you'll see that we have these in IntelliTip. So if you switch from looking vials to looking through packaging, you just take the tip off, it automatically loads the correct parameters and such. So that makes the usability really top notch. This is a class 1 laser safe product, so you don't need a laser safety program, you don't need safety goggles or anything else. And so this is really important for being able to use this in any environment. And again, the graphical user interface known as the GUI is Opus Touch, the same as on our Alpha product, so you don't need to learn two different software interfaces. And again, you have the verification identification workflows. Maybe most importantly is it has very, very high wavelength accuracy, better than one wave number, and typically it's usually around 0.2 wave numbers. That's better than most lab systems that are out there. It's really important because remember, you're going to be searching databases for unknowns and databases again to verify. And the number one criterion used for those database searches is wavelength position or peak position. So this is really important. So in a validated environment, you don't have to worry either. About a third of all, I think this number is actually about half now, of all the largest pharmaceutical companies are already using the Bravo. It was developed for looking at incoming raw materials ID in the pharma industry. And again, it's compliant with all pharmaceutical regulations. And you'd say, well, what if I'm not working in a pharma environment? It's still good. All of these compliance regulations, the system constantly testing itself, constantly making sure the wavelength is good. All of these things certainly meet or exceed the GMP, GLC, and GCP compliance for doing pharmaceutical work. And as the system is 21 CFR Part 11 compliant. But that also means that it's very reliable for even an analytical lab, that you don't have to worry about the system. If there is a problem, it will let you know what the problem is and what you need to do. Now, I mentioned wavelength precision. So the system uses what we call SureCal technology. So that means that the wavelength is always going to be very precise all the time, better than one wave number precision. And so very important for quantifications and for artifact-free spectral subtractions. Here's a good example of where we're doing the subtraction on the left without the SureCal and on the right with SureCal. And you can see how nicely we get rid of any artifacts that might be there with any drift in the wavelength precision over time. And that's due to the active control of the laser, and we also constantly monitor the laser. So this is how the calibration works for the Bravo. We have a built-in neon reference lamp, which is patented by Brooker to use a calibration standard inside the spectrograph. And then we monitor the emission from the neon lamp all the time with the Raman signal, such that the wavelength is calibrated to the best possible wavelengths. Other features I mentioned before, we actively shift the laser 3 times each laser, and that enables us to get rid of the fluorescence and maintain sensitivity that you wouldn't have if you tried to move to even longer wavelengths at 1064. So here's a good example looking at talc and polyethylene bags. And you can see we shift the laser 3 times, and now we can divide off the fluorescence and any absorptions that you might have and get a perfect spectrum of the talc. This is really unique to any handheld system to be able to collect this quality data with getting rid of the fluorescence so efficiently. Here's a good example of lactose monohydrate. And again, normally this is very challenging to collect good data with, and yet you can see that in just a few seconds we can collect a beautiful noise-free spectrum. You'll also notice again that the baseline is good, and even these very small minority bands can be readily resolved. And again, that has to do with the signal-to-noise performance of the system also. So with the duo laser configuration, we use 2 lasers at the same time so that we can have the response that's optimized for the data acquisition. You can see for acetamidophenol that we can collect a beautiful spectrum without having to go to 1064 like you would traditionally have to with FT Raman. Here's a host of others. So you can see EDTA, potassium acetate, glacial acetic acid, CHAPS and tween 20. And even you can't get these normally at 785 or 852 that we normally might try to do these at. And even at 1064, you can see the result is really poor. Yet with the Bravo, you can get really high quality spectra at the wavelengths with good signal to noise and good just really reproducible and quantifiable results. I mentioned that it's a class 1 laser safe product. This is really important, such that you don't have to worry about damaging the sample or damaging your eye. Most handheld devices are considered class 3B devices, which require safety programs and safety training and everything else. None of that is required with the Bravo. I mentioned the Intelli tips. This is really nice that we have an encode for each tip, so you just take the one off that you don't need. Sometimes for our art customers, they want to make sure that they're putting as low power as they can on, and so we have adjustable focal point tips available as well. You can, we have customizable tips for doing tablets and other types of products, as well as doing vials. So again, you just screw the tip off and put the one you want on there, and away you go. This is the workflow, the GUI interface that I mentioned. So you have the Alpha 2, which is our portable FTIR, and our research FTIR platform, the Invenio. Again, both use the Opus Touch platform, and with the Bravo, again, using the same GUI. And then the workflows, I think software is as important as hardware in these cases. So when we look at verification workflow, we want to make sure that you can get exactly the result you want without complicating it. So this should be red light, green light, or pass-fail operation, in which it certainly does. And then with the identification workflow, you identify your unknowns. And again, you can give the samples a name and other important details such that you can know what you analyzed. So very nicely done. I also mentioned the compliance. So you have the data integrity is maintained. It's a GMP compliant platform and complies with all US Pharmacopoeia regulations and European Pharmacopoeia regulations. I mentioned wavelength accuracy. So you can see this is a typical installation of a Bravo, and you can see right where my pointer is that essentially it's about 0.2 wave number wavelength precision to the ASTM cyclohexane reference standard. And lastly, it just works great for connectivity. So I usually have it wirelessly connected to my laptop, and if I want to do the acquisitions on the laptop, it works great, and the data is transferred seamlessly back and forth. If I acquire the data on the handheld, again, later I can transfer very freely. This is the most flexible platform because, say I go into an art gallery or a museum, sometimes I just want to do the acquisitions using the handheld, sometimes I want to use the PC. You can do either or, they're interchangeable very nicely at any point. And in summary, this is a unique device designed from the ground up to address historic shortcomings in Raman instrumentation. You can get rid of fluorescence in almost all cases. It's a class 1 laser safe product, so you don't have to worry about using it. It's very, very easy to use for both verification and identification of unknowns, and it is very accurate on the wavelength axis so that you can get the right hits in the databases so that it is effective in doing those verifications and identifications. And with that, I thank you for your attention.
Bravo, Tom. Bravo. If you've got any specific questions for Tom, or indeed any of our speakers today, please simply click the Ask a Question button. It really couldn't be any simpler than that. Okay, our third presenter is Moritz Fisher, who's a sales engineer with Hamamatsu Photonics Europe, and he's going to help you design the optimal process analytical setup, amongst many other things. Moritz, over to you.
Welcome to today's presentation, and thank you for tuning in. My name is Moritz Fisher. I am working as sales engineer at Hamamatsu with a focus on analytical applications. Without a trace, or rather with a trace, that's the slogan of this webinar, because we will be discussing Raman Spectroscopy, a very distinct kind of spectroscopy, which can be tough to implement due to its signal strength. You could in some way call it detective work, because it can be difficult to acquire the Raman signal in a good manner. But at Hamamatsu, we are optimistic to help you with it. Even though the extraction of Raman signals can be a hurdle, there is a high chance that you earn quite some glory by its implementation. A plethora of information is actually hidden within Raman signals. First of all, why is Hamamatsu discussing Raman applications? Thanks to our long history and our strong network, we have provided many different industries and applications with high-quality optical components, and we want to share some insights with you. We take a large pride in being a one-stop shop in the photonic and optic industry, and we have a close cooperation with a large number of OEMs who create photonic and optical devices and systems themselves. We were founded almost 70 years ago, and since then we have grown and advanced our technology ever since. In my humble opinion, there are two major aspects which make Hamamatsu as a company unique. First, we are present and in direct contact with our customers all over the globe. And second, we put a tremendous amount of effort into research and development. That means we spend approximately 10% of our profits back into R&D. And our customers benefit greatly. Not only we have a wide lineup of standard components, but we are also always open for discussions of a product with customized specifications. So in the meantime, we have more than 15,000 officially registered products. And those are, for example, lasers, light-emitting diodes, xenon lamps, deuterium lamps. And on the sensor side, we have photodiodes, linear image sensors, two-dimensional image sensors, and so on and so forth. It even extends to X-ray products. One big trend in all photonics applications is Raman Spectroscopy. Its share is steadily growing in various analytical measurement problems. For example, being a non-destructive technique, Raman Spectroscopy can analyze historical art if you want to check its authenticity. While regular, visible spectroscopy will analyze the color only, Raman Spectroscopy can also check the crystalline or molecular structure of the paint. In a similar manner, Raman Spectroscopy is an excellent tool to check crystalline samples, for example in relation to semiconductor materials. Lastly, Raman Spectroscopy can also be a great tool in the chemical and pharmaceutical industry because it is a point-and-shoot technique without the requirement of sample pretreatment. Those examples were just to name a few. So, as you can imagine, Raman Spectroscopy can be a powerful and non-destructive analysis technique with a variety of applications. That is why, in our opinion, you should be familiar with the technique itself. But actually, it is not trivial to record those kind of high-quality spectra. On the contrary, it is actually quite a challenge. The share of Raman scattered light is actually ridiculously small. It is very tiny. That means the majority of the light is Rayleigh scattered, so the outgoing photon energy is the same as the ingoing photon energy. To put it in terms of numbers, the probability of Rayleigh scattering is larger by factors of millions to billions. Or to put it in other words, you could compare it to the measurement task of seeing a coin on the Mount Everest, and you do it with the same measurement instrument as measuring the height of the Mount Everest. That is why I like to compare it to a detector's work if you want to nicely resolve and capture those Raman signals. So the physical properties of the Raman effect leave us with many challenges. The Raman signal itself is very weak and faint. Furthermore, you need to have a very high spectral specificity in order to resolve narrow features. Then there is the problem of the excitation signal. That means the Rayleigh scattered light heavily disturbs the Raman signal. And also, fluorescence signal needs to be filtered out, or taken into consideration at least. And lastly, dependent on the signal-to-noise ratio that you want to achieve, you can also have to face large integration times, and it is possible that you need a whole lot amount of averages. On the other hand, Raman spectroscopy also offers plenty of chances. First of all, the kind of signal generation is very distinct. So if absorption or fluorescence spectroscopy do not work out for your measurement task, you can simply try Raman. In addition to those other methods, there is also detailed structural information that you can win with Raman spectroscopy. Furthermore, there is no sample pre-treatment needed, and also the design flexibility is large. As I have already said, you can choose from a selection of excitation wavelengths, for example 532, 785, 1064 nanometers. You can upgrade or you can use it within confocal microscopy setups. You can perform imaging with microscopy setup. And lastly, it is also upgradable to SIRS. In Raman spectroscopy, SIRS is the abbreviation for Surface Enhanced Raman Scattering, or Raman Substrate. This technique is an extension to regular Raman spectroscopy if the signal strength acquired with regular Raman spectroscopy is not sufficient. In this case, one employs a substrate which has a metal structure on the substrate. For example, those can be silver or gold nanoparticles. As a consequence, the excitation light will interact with the metal substrate and the sample, which is placed as a solution on the sample, on the substrate. And then a physical effect will take place, which is called Plasmonic Interaction. This will enhance the Raman signal by multiple orders of magnitude. So the range for this amplification is 10 to the power of 3 up to 10 to the power of 6. As an example, you can have a look on the chart shown. It shows a spectrum of a bipyridal solution, first on a regular glass substrate as the bottom spectral line, and second, the same solution on a SIRS substrate. But please have attention that the concentration of the solution on the SIRS substrate is lower by a factor of 100. As you can clearly see, the solution is nicely resolved in the Raman spectrum if the SIRS method is used. So as a take-home message for this method, if you have no success with regular Raman spectroscopy, you can also try with the SIRS method as an extension. Next up, let's have a look at how a Raman spectroscopy setup could look like. What are the basic components? Of course, a laser is needed as source. You can select a wavelength and a laser power of choice in this case. Next, you need to take care of the whole optical path. This means you need to have suitable mirrors, gratings, filters, and so on. The next key component is the sensor for signal acquisition. For this, you can either use a linear image sensor to capture the spectrum, or you can use a whole ready-made mini spectrometer. And lastly, to round this off, you need the appropriate circuitry and software. If you combine all of this together, you obtain a whole Raman module, which you can use for measurement. At Hamamatsu Photonics, we have developed many different styles of sensors and spectrometers for Raman applications. These can do the detective work in your applications. So let's have a look at some components on different integration levels. First of all, there are complete miniaturized Raman modules in our lineup. This means they are complete mini Raman systems ready for measurement. Those integrate a small laser diode. They include all of the necessary optics. They have a sample holder for a regular or a SIRS substrate. And they have a spectroscopic sensor inboard for signal acquisition. Next up, there is also the signal processing software on board. So you simply need a USB cable to display the data. This makes such kind of device a very flexible and powerful tool for mobile Raman measurements. Generally, we are quickly able to adapt to specific measurement tasks, and that is because we mount our own mini spectrometers in those Raman mini modules. Those mini spectrometers are also a great starting point for the development of a compact Raman spectroscopic system. The slide you see now shows our standard lineup of mini spectrometers, which are suitable for Raman measurements. So that means they were developed as spectrometer with focus on high sensitivity and a fine spectral resolution. They simply communicate with a computer via USB, and they are fiber coupled. That means they are an easy-to-use turnkey system which you can readily integrate in your spectroscopic system. We can help you by designing a customized spectrometer if you have a special wish, a special specification in terms of the spectral resolution or also the signal-to-noise ratio. Lastly, if you want to have the full design flexibility for your Raman spectroscopic system, I advise to choose a sensor on a component level. And there is a wide range of suitable sensors for this. On the acquisition side, there is also a trend towards acquiring Raman spectroscopy signals with spatial resolution. As Raman spectroscopy needs high sensitivity, in turn, particularly laser scanning microscopy can ensure a good signal quality. In this case, we can recommend you to have a look at our microscanning mirrors, which are based on our microelectromechanical technology. In a nutshell, if you want to perform Raman measurements or if you want to design an OEM system, there is a whole variety of different points where you can start from, ranging from a whole miniaturized portable Raman system down to single spectroscopic line sensors. But what could their implementation look like in reality? Let's have a look on cases of Raman spectroscopy which are already solved. First, to get back to the starting slide, Raman spectroscopy can be useful in the beverage industry. For example, in the determination of ethanol and methanol. You can have two applications in mind here. For instance, in a quantitative measurement, you can probe the content of ethanol, so drinking alcohol, in beverage production. This way, ethanol content can be precisely controlled, and you can guarantee that you have a homogeneous product all of the time and all over the world. In the example spectrum, you can see the Raman spectrum of an alcoholic liquor in dependency of the ethanol concentration. So you can nicely see that you can calibrate the ethanol content with it. On the other hand, we can also have a look at qualitative measurements. In 2008, in China, there was a large scandal which was about milk and milk powder. In this context, dairy companies intentionally added melamine, a nitrogen-rich synthetic organic material, to their milk and milk powder products. The reason for this was that the high melamine content fooled you to think that the milk or milk powder had a high overall nitrogen content and hence protein content in laboratory analyses. So this way, dairy companies wanted to use less amount of expensive genuine milk. However, this had heavy consequences on the human kidney upon consumption. Some children even died. So this proves that there is a necessity to detect food adulteration in a portable manner. And the suitable solution for this is, again, to use a portable Raman module in combination with source substrates, in the combination with milk powder and melamine. In the chart shown here, you have an example measurement of melamine in solution. It shows a comparison of melamine on a source substrate in comparison to melamine on a silicon substrate. Again, please take note that the concentration with the source substrate is lower by a factor of 100. And as you can see in this example, the Raman module with the source substrate can detect the melamine and can hence be a portable device which guarantees the food safety in regard to adulterated milk. In terms of content, I am finished at this point. I hope that this webinar gave you a learning about Raman spectroscopy in general and how you can implement it with some basic sensor components. If you have any questions or any sort of requests, please do not hesitate to get in touch with Hamamatsu or also get in touch with me directly.
Many thanks, Moritz. Very illuminating. If you'd like Moritz to tackle your specific analytical need, now is the time to submit your questions for the discussion later after our final presentation from David Nichols, a Senior Applications Engineer at DRS Daylight Solutions. He's going to shake things up by stepping away from Raman and into the infrared with a presentation on technology stolen straight from the Star Trek Enterprise, probably, quantum cascade lasers. David, the floor is yours.
Hello, my name is David Nichols. I'm a Senior Applications Engineer for Daylight Solutions here in San Diego. That means I get to work with our customers to help them do amazing things using our lasers and sort of share along with their progress in scientific fields. Today, I'll be talking about how quantum cascade lasers, or QCLs for short, provide solutions that benefit established and emerging applications in environmental research. Specifically, we'll focus on microplastics analysis, environmental monitoring, and water quality analysis. Quick introduction to Daylight Solutions. Daylight is a leading provider of unmatched field-proven instruments. We have a portfolio of IR sources addressing a myriad of molecular sensing and spectroscopy applications for OEMs and researchers. We have over 15 years of experience fielding laser sources and instruments for environmental and other applications. In this presentation, I will review some background on the application's demands, speak briefly about QCL technology, and end with a few application examples where we actually apply this to the environment. So what are the current challenges in environmental applications? Well, there are many of them. Like many spectroscopy-based applications, many environmental monitoring applications benefit from rapid data acquisition. This is especially true if the analysis of many samples is required, as is often the case in something like microplastics. Rapid data acquisition allows higher signal in any given measurement time window, helping to improve the signal-to-noise ratio. High signal-to-noise ratio for highest quality of data, and especially in areas like remote detection, helps us in applications like measuring pollutants and detecting multiple molecules of interest. So how does Daylight's portfolio of instruments help this out? So with over 15 years of working in environmental applications, we've worked to identify these technical challenges and develop technologies that address the current demands of environmental research, making our products faster with higher sensitivity, higher beam quality, and extending the wavelength coverage of our QCLs. So Daylight Solutions works primarily in the mid-infrared region, about 3.3 to 13.7 microns. Many of you are likely familiar with this range, but let me highlight a few advantages. First, the mid-IR range is highly sensitive to most molecules, making it an ideal range for surveying many samples, either known or unknown. You can see on the right that many molecules of interest for environmental applications are detectable in this wavelength range. So working here shows us the things that we're actually likely to be interested in. Additionally, mid-IR is highly selective for multiple species, allowing multiple molecules to be detected simultaneously. This is why the mid-IR region is often referred to as the fingerprint region for its ability to ID several unique chemical signatures. So historically, there have been many mid-infrared sensors that are commercially available, but these sources lack the spectral brightness and power to really address the current applications. So QCL IRs, what's so wonderful about that? Well, at the core of our technology is our external quantum cascade laser, or as we often refer to it as QCL-IR. Quantum cascade lasers were first realized in 1994 at Bell Labs. Daylight was a pioneer in commercializing this technology in 2005 and actually bringing it into the marketplace. The high spectral brightness of the QCL source is orders of magnitude higher than commonly used mid-infrared sources, such as the globar filament used in FTIR systems or the light emitted by a synchrotron facility. And the plot here shows the spectral brightness as a function of wavelength for these various sources of infrared light. And notice that this vertical scale is logarithmic, so several orders of magnitude brighter. We care about this because it enables a higher signal to noise ratio to produce the most reliable data. Starting in 2005, Daylight expanded its core business of QCL sources into an expanded portfolio of laser sources from high power, MOTOP-free, narrow line width solutions onto multi-channel broadly tunable lasers. For the last 7 years, we have integrated these sources in complete end user systems for QCL-IR microscopy and real-time QCL-IR for liquid analysis. So we make both the lasers and then we put those lasers in instruments to make them easier to use for experimenters. So today I'll be talking about a few established and emergent environmental applications enabled by QCL technology. So environmental monitoring, water quality analysis, and microplastics analysis. So Daylight has had 15 years of experience developing molecular spectroscopy applications, and this includes environmental applications. Remote standoff detection has been at the forefront of air quality analysis. For instance, in collaboration with the Merth Center, Princeton University, and several other academic institutions, Daylight Solutions deployed a QCL open path system at the 2008 Beijing Olympics to measure ozone, ammonia, and carbon dioxide. In other words, your significant atmospheric pollutants. This system was placed near other systems already in use to measure air pollution. The QCL-based system showed results comparable to the then existing technology, but it offered more continuous measurements. In other words, you could take your measurements as fast as you could sweep the laser. Notice that you see the diurnal variation. If you look at the plot of the concentrations of the pollutants, you can see them change with time. And notice that the sensitivity goes down to a low of less than a part per billion. So this is a very sensitive measurement of what you actually have in the atmosphere. Daylight has continued to offer products with higher signal to noise ratio, lower beam pointing, and a narrow line width to make remote detection of chemicals feasible. Our customers have also put our lasers to use in other gas sensing applications. So this is taking a laser, you make it part of your experiment, you take advantage of the nice light that we create and the fast scanning that this enables, and you can look at things like measurement of greenhouse gases and shock tube gas sensing. So turning from gases to liquids, water quality analysis is a big challenge. So heavy pollution creates new challenges for wastewater treatment facilities and a demand for new methods to accurately identify and quantitate pollutants. Regulatory bodies publish guidelines on the maximum allowable contaminant concentrations in drinking water. However, current methods for assessing water quality require multiple manual sample preparation steps that make real-time water analysis impractical. Infrared is often not considered for analyzing aqueous samples, given that water is a strong absorber in the infrared. And the plot here shows the absorption of water through our mid-infrared region And notice it absorbs quite heavily in the specific areas that we're interested in. So as we'll show in the following slides, QCLs can overcome this absorption given the high spectral brightness of our lasers. So this is our Colpeo liquid analyzer, which uses QCL IR to provide real-time analysis of aqueous solutions. So you have real-time high sensitivity measurements that take advantage of our high spectral brightness QCLs. The high power of the QCL source, coupled with its broad tuning, enables high sensitivity through relatively long path lengths with a large dynamic range. So the Colpeo liquid analyzer is a robust analytical platform that can be used for quantitative analysis to look at levels of organic and inorganic solutions. So this is a lab example of data from the Colpeo looking at concentrations of ammonium chloride, where ammonium and its friends are a fairly interesting water pollutant. And you can see the distinct signatures, and you can also see that it responds linearly to the concentration. In other words, we can not only identify that the substance is there, but we can tell how much of it there is in the liquid, and do this all in real time. Here's another example of using QCL IRs for continuous water quality analysis. This is from Max IR Labs, where you're looking at the concentration of nitrite, nitrate, and ammonia in water. So each with its own distinctive spectral absorption peaks. Microplastics. Microplastics are defined as very small pieces of plastic that are less than 5 millimeters long. While it's known that humans and other species can absorb microplastics, the effect of microplastics are not yet fully understood. As plastic pollution increases, so does the urgent need to understand the potential risks that microplastics represent to living organisms. But we need data. So the analysis of microplastics and nanoplastics is a rapidly evolving field that aims to understand their effects on the ecosystem and help solve the global plastic pollution problem. Detecting and analyzing microplastics in the ecosystem is a critical part of this effort. Infrared spectroscopy is emerging as a powerful technique for the sensitive detection, identification, and imaging of microplastics. So here is an example of data taken with our Spiro microscope by experimenters at the Alfred Wegener Institute. So they leveraged our label-free QCL-IR imaging to classify microplastic contaminants in waterways. The high throughput detection and broad wavelength range made the Spiro microscope ideally suited to distinguish between contaminants at a rapid rate. So if you look in the upper right of the slide, those are images taken from the Spiro. Each of the little blobs there is a piece of microplastic, and you can look at the spectra of each of those and use the spectra to determine what kind of plastics they are. So using the Spiro allows us to determine the size, the chemical composition, and the shape of each of the pieces of microplastics in the sample. So we'd like to collect a lot of data, which means we'd like to automate the microplastics workflow. So we collect samples from snow, soil, waterways, dust. We place them on a barium fluoride slide, put it in a sample holder, put it in the Spiro, and the Spiro can take a scan of one image in about a minute. So you get very rapid throughput on that. And then we pass the data on to a chemometrics program to figure out what kinds of plastics they are. There are also other applications that people have used the Spiro for. So for instance, in the bottom, in the middle, experimenters at NIST looked at thin membranes, and they were interested in using the polarized light of the laser to study the different domains. So the fact that you can select the polarization orientation is a great advantage there. And this lets you study membranes as they're laid down, and also in the microplastics realm lets us study the change in the structure as time goes on. So thank you for your time. Please contact us if you're interested in this or you have further questions. Thank you.
Thank you very much, David. Fascinating technology enabling real-time measurements in some increasingly important application areas, I think. It's now time for our live question and answer session. And I've got some good news and some bad news. Do you want the bad news first? Hamamatsu's Moritz Fisher is not able to join us for the Q&A session. However, the good news is that he's coerced his colleague, Christoph Vernal, who's Senior Sales Engineer Analytical at Hamamatsu, to join us instead. So, presenters, are you all ready, willing, and able for some questions from the audience? I'm getting nods and thumbs up. This is good news. Great. So first question, Miriam, you've been sat there waiting since the beginning, so I'll field this first one to you. They ask, can particles in liquid be imaged with the confocal Raman microscopy?
Basically, yes. Some of the samples I've shown you, the cell samples, for example, have been measured in buffer solution. Raman images typically take minutes or at least seconds to be acquired. So your sample or your particles should be fixed inside. Or if you measure in liquid cell, you can see what signals come by, but then you don't get the image. But you can measure in liquid, yes.
Fantastic, thank you. One for Tom. I'll pass this on, Tom, but you may want to respond outside of the forum. But just so you know, someone asks, says, great talk. How costly are these handheld Bravo instruments? We'll dig that one out for you. But an actual question. Another person asks, can you say a little more about the type of algorithm you use to match the spectra?
So the first one, you're correct. The salespeople like to answer those questions rather than the science people. So I'll defer to that, to that one. But typically in the U.S., it's about 50,000 plus or minus. And the algorithms that are used, most database searching algorithms, and this is true probably globally, are using peak positions more than anything else. So there are other clever secondary criteria that are used, but in general, the peak position is most important.
Great, thank you. One for David. Being as we finally managed to get you onto the back onto the call, they say in your talk all the laser sources and instruments you mentioned are widely tunable, but they see fixed or narrowly tunable sources used in some environmental applications. They say, can you comment on the pros and cons of fixed or narrowly tunable versus widely tunable sources in environmental applications?
Well, so Daylight sells both widely tunable and fixed or narrowly tunable lasers. The advantage of the narrowly tunable one is if you have a single spectral line and you'd like to see how it changes quickly with time, you could just sit on that spectral line. The advantage of the widely tunable ones is that you can, if you have a mixed sample, you can acquire an entire spectrum fairly rapidly. So our Meerkat can sweep across its entire range in under a second. And then you leave it to your algorithms to try and deconvolve the spectra you get so you can pull out multiple components in the sample.
Great, thank you. Christoph, one for you in place of Moritz. They ask, this is a SIRS question. They say, do I need to use a SIRS substrate or can I use the module without it?
So there's no need, well, in principle, to use the SIRS substrate. You can also measure the Raman effect without a substrate. It's just it's very easy to unmount the sample holder, and then you can do it in free space. However, depending on your application, it might be useful to use the source substrate because, well, as shown, the source effect enhances the Raman signal.
Excellent, thank you. And while you're there, obviously you showed many different products, or rather Moritz did. They say, could I get the spectrometer into another shape to fit my setup?
Sure. It is possible to customize the spectrometer so we can easily integrate it into your device. Also concerning customization, you could also have higher laser powers, like for example, 50 milliwatts, or you can change sample holder for another shape to have qubits or something like that.
Great, thank you. One for Miriam. They ask, can I also measure rough surfaces without being out of focus? And if so, how much height variation would be okay?
Yes, we do have a technology. This is hardware edition, which allows you to follow rough surfaces. So this hardware measures the distance between your objective and sample and simply keeps the sample in focus during your Raman measurement. So this is independent from the Raman measurement and allows you to follow the surface. And it's not really limited by the roughness of your sample. Actually, the maximum travel range that can be covered by our system is 3 centimeters. So typically the samples are much flatter. And then it's, yeah, as long as it's not a 1 millimeter step, you can simply follow the surface. And this is important because in confocal Raman spectroscopy and microscopy, we have a very high spatial resolution. So it's definitely important to keep the sample in focus. Yeah.
Great, thank you. Complex one for you, David, or at least it seems complex to me. They say, do you find that coherent artifacts limit spatial resolution in your Spero QCL IR microscope? And how do you handle these?
Yeah, I'm going to follow like Tom did with the price question. So we have proprietary techniques that basically break up the coherent artifacts. So we are able to get down to essentially, we're limited by the wavelength of light, infrared light. So we're basically down at the diffraction limit, so we can image styrofoam beads, say down to 10 microns in diameter without much trouble.
Thanks so much. One for Tom. Are there any databases available for subscription for Raman spectral searching?
Yeah, this is one that is kind of difficult. When you look at infrared spectroscopy, there are many databases available. You can find them almost anywhere. For Raman, the databases have lagged behind. So Wiley and others offer databases, including Brooker and probably everybody that sells instrumentation, sells databases along with them, either directly or third party. But sometimes you don't know what you're looking at. So if you're characterizing knowns, that's easy. If you're doing quality control and you have an unknown, that can be more challenging. And sometimes a subscription makes more sense, where if you search an enormous database like Wiley would offer, then you can, you just pay a small amount for the hit that you end up getting, and you don't have to have the reserve of all, tens of thousands of spectra that you paid for that you will never hit.
Thanks so much. Here's a long question, so bear in mind, and I guess this is open to anyone really. They say the biggest disadvantage of IR or Raman has always been that you need reference spectra and spectral calculations are not advanced enough to help. With MALDI and nano SIMs now outperforming Raman and IR imaging, with the added advantage of being able to ID components without references or bulky labels, where do you see vibrational spectroscopy imaging evolving to still play a role in the lab? Miriam, do you want to take a shot at that one?
I can make a start, sure. First thing that comes into my mind is that Raman vibrational imaging is not non-destructive. So you do not damage your sample. You can further use it. You can further analyze it. You can put it to any kind of environmental control or environmental conditions, so you can measure in solution. You can use heating, cooling stages to see phase transitions. You can image dynamics as you do not destroy your sample. You can even put the system in a glove box. Maybe the others want to continue, but there are so many things actually, and so many applications where you even do not need the database because you know what you have and you just want to get the spatial distribution of your components. So not all applications do require databases.
I actually consider them complementary. Brooker offers a wide range of multi-imaging systems, and we found that even in the art community, that where people have infrared and Raman imaging systems already, that multi-imaging is really quite useful. Miriam's correct that you do have to consume some of the sample to actually do the mass spec. Usually it's not much on the surface. And I like to think that infrared and Raman imaging are kind of front of the line techniques, and then you can move to SEM or mass spec imaging. And I think the information is very complementary. So I think that if you have the resources, I would, in my lab, I'd like to have both. And in fact, in our demonstration lab, we have the Rapidflex that is 50 feet away from our infrared and Raman imaging systems. And so, yeah, I think you want to use both. There's technical merit for both.
There's also sometimes application, like for example in plastic sorting, which you do with hyperspectral imaging. There is a big advantage this you can't do with mass spectroscopy, not in this so exactly, and not in this needed speed that you will use when you have to sort plastics.
Thanks so much. Any comments on that, David, from you?
I mean, the one thing is, if you are in, for instance, an industrial manufacturing environment, the Spiro can take, a scan across its entire range in under a minute. So if you're doing quality control stuff, you can do it non-destructively, you can do it rapidly, and you can sort of map out what the chemistry is there and how it's distributed across your object.
Thank you very much. Specific one for you, Miriam. They ask, what kind of sample preparation is required for RISE experiments?
RISE, it's the combination of SEM and Raman. You have to be a bit careful here because for SEM, you typically sputter your samples. You should not do that because a metal film will hide your Raman signal, and a carbon film, the carbon is very Raman active, so it will dominate your Raman spectrum. So what we typically do is we first measure the Raman and afterwards the SEM image. And yeah, usually you work in low vacuum to avoid charging then. Besides this, no specific sample preparation is needed. Just a rough, at least roughly flat sample is advantageous.
Thanks very much. One for you, Tom. They say, how long does it take to sample something at 2 excitations and process those with high fluorescence? And is this approach also available in a benchtop instrument by Brooker?
Yeah, so that's a good question. So we actually do not offer the shifted excitation on our benchtop system. We would like to, and maybe we will in the future, but we don't right now. It's very fast, so we directly control the both excitation lasers on the Bravo. So it literally takes just a few seconds to collect 6 spectra total, 3 at 785 and 3 at 850.
Thank you very much. Christoph, I'm not fully sure I understand this exactly, but I shall ask the question anyway. They say, is it possible to evaluate the spectrometers? Seems like a very broad question, but shoot.
So I think, well, it maybe means we do have demo samples that we can loan to the customer. So you can just contact us and get the spectrometer to test. If this is the question, I think so. Next year, there will also be a release of a higher source Raman module with higher power and extended range for the Raman shift. Also, concerning the software side, we do have a new software from our side that supports all the Hamamatsu mini spectrometers, and you can even operate multiple spectrometers at once. So this makes it much easier for the future. Just contact us and we will supply you the software.
Thanks, Christoph. I think this one is for you, Miriam. They say, given the relatively weak Raman effect, what kind of sensitivity can be expected from confocal Raman spectroscopy compared to confocal fluorescence spectroscopy?
This is a good one. Yes, the signal is weak. Well, first of all, this led to the fact that first people didn't use Raman that much, and then they improved the hardware quite tremendously. So nowadays, the Raman systems are highly sensitive, which means, well, there are two reasons why you want to be very high sensitive. You want to measure small particles or low concentrations, and you can do that with a confocal system. You can get signal from nanoparticles, or as also the colleagues from Hamamatsu introduced, you can use SIRS substrate to enhance the signal and push it if you want to measure low concentrations. Or if you think from the other point of view, you want to speed up your measurement. So you want to increase the sensitivity to get a sufficient signal to noise ratio in a shorter amount of time. And if your signal is high enough, like for inorganics, polymers, graphene materials, then we can do imaging with integration times of less than a millisecond. This has been made possible by hardware. So you can really speed up your measurement. Sensitivity is high enough.
Great, thanks, Miriam. I think we've got time for a couple more. One for you, Tom. I don't know if this refers to the laser certification that you mentioned, but they say, do you need to have a safety program to use portable or handheld devices or probes?
So if the portable handheld devices are classified as level 1, then you don't need to have a safety program. That's really important because if you have to have a laser safety program, that you have to have a laser safety officer. You usually want to have internal and external audits associated with that program. So there's a huge burden that goes along with that. So when we developed that product, we really wanted that to be a class 1 laser safe product. The same as if you were checking out at the grocery store.
Great, thanks, Tom. And perhaps one more for you, Miriam. They say, what is the achievable spatial resolution of confocal Raman systems? And what's the minimum size of features that can be investigated?
Well, this is actually two different questions. Confocal system, if you're truly confocal, that means you can reach the diffraction limit. We are working with visible lasers typically, so we are talking about a lateral resolution of 200, 300, 400 nanometers, depending on the wavelength. In Z direction, so the axial resolution is a bit less. In confocal systems, nevertheless, it's still 1 micrometer. So you can measure inside transparent samples with a depth resolution of about 1 micrometer. You can measure smaller particles. We touched this topic before. Because sensitivity is high enough, you can even measure smaller particles, like nanoparticles. With SIRS, I guess also single molecules, for example, can be measured.
Fantastic. Well, thank you. I'm afraid that's all we've got time for today. I'd like to thank all of our experts once again for their presentations and contributions to the question and answer session. I'd like to offer a big thanks to our sponsors, Ytech, Brooker, Optics, Hamamatsu Photonics Europe, and DRS Daylight Solutions. And finally, I'd like to offer a big thanks to everyone watching. So thank you again from me, and goodbye.